Okay, we're going to start our lecture today on the evolution of sex and comparison of sexual reproduction and asexual reproduction from an evolutionary perspective. Okay, so let's start with a clicker question here. So, go ahead and take a minute and think about non-random mating. How many examples you can come up with here? We'll talk about it later. Okay, so one really important form of non-random mating is inbreeding. So this violates one of the assumptions of Hardy-Weinberg, which assumes random mating. Okay, this can affect genotype frequencies without affecting allele frequencies. And so think about what other processes can do this. And in particular, we'll talk about one form of inbreeding called selfing, or self-fertilization or self-pollination if we're talking about plants. Okay, this is not the same thing as cloning, which is a somatic division. Or in plants we think of clones as vegetative clones, where like you have root suckers growing out and they start a new shoot. That's not the same thing. So selfing is the ability to self-fertilize between sperm and eggs, or self-pollinate if you're talking about pollen and ovules in a plant. Okay, so lots of plants can do this, and some animals can. So the key here is that homozygotes will always give rise to homozygotes, and that just comes straight from your Punnett square. Big A times big A yields all big A. And the same for little a. However, with heterozygotes, so if you have big A times big A, you produce half homozygotes and half heterozygotes. Okay, so that comes straight from the Punnett square. All right, so you have a 1 to 2 to 1 ratio of this genotype, this homozygous genotype, to heterozygote to the other homozygous genotype. Okay, so what's the problem here? Well, if you have a population of selfing organisms, you can only breed with yourself. So you can only breed with the genotype you have, basically. So if you're a big A, you produce big A. So it's just a Punnett square, right? If you're a little a, you can only self, so you can only reproduce with little a, which is yourself. And the same is true for the heterozygote. But here's the important part here. So even if you start out with a population at time 0 with 100% heterozygotes, 50% of the offspring that you produce are going to be homozygotes every generation. So even with 100% heterozygotes starting out, you produce 50% heterozygotes and then 50% homozygotes. So 25% big A, 25% little a. That comes straight from a Punnett square. If we go to the next generation, so S1 to S2, remember, we're a selfing population, so these big A homozygotes can only reproduce with themselves, so they can only produce big A. So they're kind of locked in the homozygote gene pool. And the same is true for the other homozygote, whereas you get the same situation happening again with your heterozygotes. So now only 50% of the population are heterozygotes, and when they go to do a second round of selfing, that reduces to 25. So you're dividing that 50% again, 12.5% this homozygote, 12.5% that homozygote, and 25% of the total population are heterozygotes now. And so what you keep doing every generation is you end up losing heterozygotes, because you can't have this homozygote crossing with this homozygote, for example. And so in 1, 2, 3, 4, 5 generations, you go from having all heterozygotes to just about 3% heterozygotes. So that's a very quick loss of heterozygosity. Okay, and this is especially true in inbreeding or selfing populations. So selfing can be thought of as kind of like the ultimate form of inbreeding. You're reproducing with a genetically identical individual, yourself, and that is similar to inbreeding in a way, where you tend to reproduce with genetically similar individuals. So the result is that every generation, the number of heterozygotes is cut in half. However, the total allele frequencies are unchanged. So remember, we have 100% heterozygotes when we start out, so 50% of the alleles are big A, 50% are little a. But if you count them at generation 5, you find that 50% of the alleles are still big A and little a. So evolution hasn't happened here in a technical sense. based on our definition of change in allele frequency, but we certainly have had a change in genotype frequency. So inbreeding causes a loss of heterozygosity, but that in itself can have a very strong evolutionary impact. But in and of itself does not cause evolution, right? There can be evolutionary consequences of loss of heterozygosity through inbreeding, through what we're going to call inbreeding depression, but it in and of itself does not directly cause evolution. There's no change in allele frequency in this case. What it does is it creates an excess of homozygotes and a reduction in heterozygotes. Okay, excess of homozygotes, reduction in heterozygotes. This can expose loss of function alleles, which are normally masked in the heterozygous situation. So if you have a deleterious or a harmful homozygous recessive condition, that's going to be expressed in the phenotype. Okay? And what this ends up doing is creating lower fitness among the offspring of relatives. Okay, so if we look at an example from humans, okay, if we look at on this axis the mortality rate among children of unrelated parents versus the mortality rate of children with first cousins or related parents, you can find that things are quite shifted towards higher mortality rates for inbreeding between genetically similar individuals. Okay, so this is just an example from humans, but plants do this all the time, and there seem to be very little consequences in some cases. So it's context dependent. So some plants, for example, require outcrossing. They can't actually inbreed. They're self-incompatible, whereas others are self-compatible, so they can both outbreed and inbreed. And so, some actually have evolved a strategy of selfing only. Okay, so a study from 1994 in a population of sparrows on a Canadian island. Okay, so there were two population crashes in 1980 and 1989 where the population size went down to 27 individuals from several hundred. And then in 1989, it went down to only 11 individuals. So huge, what we call population bottlenecks here. Okay, so the inbreeding coefficient of the survivors, i.e. those that were still alive in the crash year, so those that survived this environmental hardship, was much lower than the average value before the crash. So what that means is those individuals that were the most outbred were the most likely to survive. So those individuals that had parents that were genetically dissimilar had a higher chance of survival in this case. Okay, so the ones who survived the most terrible winters in this case were least inbred. And in this case, inbreeding lowers your survival chances when the environment goes bad. Why is that? Okay, and so this is going to be like, this lecture is going to explain to you so much of your life, why sexual reproduction is so important, why you behave certain ways, why you go to the gym, why you wear makeup, why you try to present yourself to the opposite or same sex, depending on who you are. So why is sexual reproduction the rule for nearly all living species? Okay, so why is it so common, and why did it evolve in the first place? So obviously every organism must reproduce to pass on its genes. Reproduction can be either asexual or sexual. Okay, it can be asexual if the cells or organisms clone themselves through mitosis. Even though we think of selfing as it's still sexual reproduction, but it more or less has the same effects as asexual reproduction. Okay, the advantages of asexual reproduction is that it does not require a mate, so you can just reproduce any old time, and it produces a perfect copy of the parent. So if you're already perfect, then why spoil it, right? We're gonna see why that's not a very good strategy. Sexual reproduction is the production of male and female gametes through meiosis. And remember, you have 2 copies of each chromosome if you're a diploid. If you're a polyploid, you might have more. And through meiosis, you have reduced gametes through down to from 2n to n. And then you recombine these gametes to produce new combinations of alleles at different loci. So offspring are of a genetic mix of the 2 parents. So you're kind of shuffling the deck every time. Okay, so if we talk about mitosis, this is a review for everybody. A diploid cell replicates each chromosome and then divides it into two cells. So both the daughter cells get identical copies of every chromosome. By repeated divisions, one cell of an organism could grow into a whole copy, and that is basically the idea behind sexual reproduction, or asexual reproduction, sorry. Okay, so let's do a quicker question, and then maybe take a break after. So how many of the following do you think are true of sexual reproduction? Okay, take a minute to do that, and then we'll take a little break. We can talk about this in recitation. Okay, break time. All right, so let's talk about sexual, I always mess these two up, asexual reproduction. So we can say attack of the clones. So these are kind of like clones of each other in a way in this case. So let's say we were making up some imaginary species of starfish that can clone itself. An asexual reproduction organism creates an identical twin or copy of itself. So a well-adapted organism, so one that's been through several rounds of selection and can survive and reproduce asexually in a specific habitat, will reproduce itself exactly as it is. This avoids the scrambling of genes, or really what we're talking about is the scrambling of alleles for each gene across many different genes. So the idea is if it's not broken, don't fix it. Okay, so in asexual reproduction, one female individual can produce 4 granddaughters in 2 generations. With sexual reproduction, the female requires a male to produce offspring. So that female will only produce 2 granddaughters in 2 generations. So the output of asexual reproduction is twice as high as that in sexual reproduction. So there's a population growth advantage. So you can have much faster population growth to take advantage of some kind of resource if you're an asexual organism. So your 2-fold faster rate of population growth means that you don't waste energy on useless males. Sorry, guys. Okay, but I'll make the argument later that maybe there is something useful about sexual reproduction after all. Okay, so what are the benefits of asexuality? So not shuffling your alleles. So most phenotypes are determined by combinations of alleles at multiple loci. So thus far in this course, we've talked mostly about a single locus with 2 alleles determining some phenotypic trait. That's the simplest case scenario, and we always use it in population genetics and evolutionary biology to illustrate the basics of, say, Hardy-Weinberg, et cetera. But if we really want to think about phenotype, we have to think about the whole genome and all of the genes involved in the expression of certain phenotypes. So you could have big A at one locus, big B little b at another, big C little c, and all 3 of those loci or genes could contribute to some phenotype. Or you could have big A, big B, little c, et cetera, et cetera. You can have all the different combinations of alleles at those 3 loci. Okay. So well-adapted organisms can clone themselves. So their good combinations of genes can stay together. So if they're already adapted, then you don't want to break up those allelic combinations that you have that allow you to survive in that particular environment, because you'll have a lower fitness. And you have a 2-fold advantage in reproduction. So when you don't have to produce males, you can just produce all females, and they can produce more and more and more. So you have 2-fold higher rate of reproduction with asexual reproduction versus sexual reproduction. Okay, males consume resources but cannot themselves make offspring. So they participate in fertilization, but they can't actually have babies, right? Seems like a waste of space, evolutionarily speaking. Sorry, guys. So the question is, why do almost all organisms that we think of in this class reproduce sexually? So when we think of some of the most diverse organisms in the world, animals like insects and things like that most plants produce sexually. So what is the advantage of sexual reproduction here? Well, sex produces genetic diversity, not in the same way that just a new mutation would, but it takes pre-existing diversity and it shuffles it around to get new phenotypic variation. So a clone of a successful parent would thrive if the clone faced identical challenges from the environment through time. So generation after generation, nothing changed. You had the same amount of sunlight, rainfall, food sources, same predators, diseases, etc. However, we know that's not the case. Conditions change from time to time, meaning that offspring may need different traits in different generations to survive. Okay, so alleles can be viewed as potential solutions to potential problems that may face some organisms. So some alleles will benefit you in one condition, other alleles will benefit you in another. And so what sex is actually doing is shuffling the deck of alleles, and it basically deals a new hand of these alleles each generation. So when you reproduce sexually, you're shuffling alleles between chromosomes because of independent assortment and crossing over. We'll talk about those here in a minute. Okay, so the Fisher-Mueller argument. All right, so to get a beneficial combination of alleles at two loci in an asexual population, so remember we're talking about two loci or two genes here, each with two alleles. So big A, big B, little b. You must have mutations happen in a sequence. So you have to have, in this case, like if your entire population starts out as little a, little b, and you know, so like why aren't you might be confused, why aren't we saying like little, you know, little a, little b. This is what's called the haplotype. And so this is a haploid organism right here. So you have one copy of the genome, let's say N. So if you have little a, little b, but you need to get to big A, big E through mutation, both of those mutations have to happen in sequence. So in one case, what we have here is, little b mutates to big B. In this segment of the population, those individuals survive and reproduce a little bit better. Little a turns to big A in this segment of the population, but at this time slice right here, over time, you still don't have those two alleles together to get you maximized fitness. So you have to wait in this case for little b to mutate again to big B. So they have to be in sequence, right? Mutation A has to happen, and then mutation B has to happen. And if you think about like chance events, which often have a very small probability, like a beneficial allele evolving through mutation, you multiply those times each other, right? So it becomes a very, very small possibility, very, very small probability when you have two or more rare events having to happen in sequence. Okay, if we throw recombination into the mix, what we end up seeing is it takes much less time to get to that optimal fitness condition of having big A, big B, because those two populations can swap alleles through sexual recombination. Okay, so if you have one of those mutations happening here, little a to big A, and then little b to big B in a separate segment of the population, those individuals can sexually reproduce, swap alleles across chromosomes, and get to this big A, big B condition much faster. Okay, so it speeds things up. So if you have to go from a suboptimal combination of alleles, here to an optimal sexual reproduction gets you to that combination of alleles much faster. And recombination is key here. That explains pretty much everything there is to know about sex. All right. Well, we'll talk about sexual selection later, which is a whole other can of worms. Okay, so you don't need to have sequential mutations with sexual recombination because you can swap alleles across individuals. Okay, so let's think about inheritance again. So how many of the following do you think are true? Do another clicker question. We'll take a break. Okay, so sex is really explained as diversity by meiosis. So crossing over between arms of homologous chromosomes creates genetic diversity by making new combinations of alleles that weren't present in either parent. And so you can see this here by, let's just color one set of homologous chromosomes blue and the other pink. Well, with crossing over, now we have new chromosomal combinations. So you can have different allelic combinations across multiple loci. Okay. So in meiosis, a diploid cell has where two copies of each chromosomes divide twice, produces 4 gametes, sperm or eggs, pollen or ovules when we talk about plants. The gametes are haploid, of course. They're the reduced gametes, and then they recombine during synapsis to form zygotes. Okay, so where does this diversity come from? So we're thinking of multiple loci here, so alleles at multiple loci. Well, the first comes from the fact that most organisms have several different chromosomes, and so chromosomes get inherited as single units. So we haven't even considered crossing over here. So we have independent assortment. So if you have dad's chromosomes here, mom's chromosomes here, you could end up with any combination of those two different forms of chromosomes in the offspring, just because they're on separate chromosomes. Okay, so that's the first level. So each gamete carries a different combination of parental chromosomes. One human egg, for example, may have 4 chromosomes from grandma and 19 from grandpa. So there's one way of shuffling variation. The other is recombination through crossing over. So let's say you have this chromosome from mom, this from dad. These are homologous chromosomes, say chromosome 17 or something. You can have a crossover event here. So you swap all the genes and alleles on one arm of your chromosomes. And then you have this recombinant chromosome here. So that's crossing over or recombination. Okay, so this will create a new combination of alleles on a single chromosome. So we have kind of 2 ways of shuffling things. We can shuffle them by entire chromosomes through independent assortment, or we can shuffle things within a chromosome through recombination. So what are twins then? Okay, so what are twins? So identical twins form from one fertilized egg. So these are called monozygotic twins. Fraternal twins form when 2 eggs are released at once and both of them get fertilized. So these are what are called fraternal or bizygotic twins. So these monozygotic twins, any twins in this class, are basically genetic clones of each other. They're genetically identical individuals, whereas dizygotic or fraternal twins are often different, more genetically different. And this is a more common situation. So about 70% of all twins are fraternal twins. Okay, so sex occurs in nearly all multicellular organisms. Why is that? So very few animal species only reproduce asexually. So that's a very rare thing in animals. A few microorganisms, so eukaryotic microorganisms like rotifers are known to do this. There's something called parthenogenesis in some fish and amphibians where females can produce offspring where fertilization is not required, so they can produce viable offspring without fertilization. So some like aphids, insects called aphids, start as asexual and then switch. And aphids can reproduce a lot of individuals very quickly. So for example, like when they encounter a new host plant, they can really ramp up asexual reproduction to cover that entire plant with little aphids. They're these little insects that suck, they basically suck the juice out of plants and make a living that way. But then once the plant is exhausted, they can switch to sexual reproduction. And of course, some plants, actually many plants can produce asexually, but some plants actually reproduce only asexually. So they are, they're self-verse or what we call apomictic, which is kind of similar to parthenogenesis. However, the fossil record shows that most asexual species go extinct. So there are certain characteristics, especially in plants, that you can use to tell whether a species is sexual or asexual. Then we find that they don't stick around very long in the fossil record. So mostly the transition goes from sexual to asexual reproduction, and that often leads to extinction. Recombination must therefore be pretty important, conferring an advantage on sexually reproducing species, allowing them to outcompete asexual species. Okay, so we've talked about the benefits of asexual reproduction. Now let's talk about the cost. So you can produce a lot of individuals very quickly, but we haven't considered mutations yet. So what do you do about mutations in asexual reproduction? Okay, let's have our starfish population here, our asexual starfish population. Let's say in an asexual population, once a bad mutation occurs, it can be passed on to all of the offspring of that line. So let's say in one of these offspring, we have a mutation called stumpy arms. Okay, so 2 of the arms are shorter than the others, and it has a selective disadvantage. And then in the next generation, we see another mutation that's harmful. That's called square tips. Okay, so now we have individuals that have both stumpy arms and square tips. So much lower fitness of these individuals that have these mutations. But the net effect is that it actually brings down the entire fitness of the population, because now you know, a lot more individuals are going to die off because they can't survive. Okay, so mutant alleles accumulate every generation. So what do you do about this? Let's say by chance, through genetic drift or small population size, all the individuals with no mutations, none of these mutations die off. So now we're stuck with this stumpy arms and square tips mutation. Okay, so bye-bye, bye-bye. Now all the remaining adults will pass on these mutant alleles, and basically things only get worse over time. And that's where the whole like ratchet thing comes in. So if you know how a ratchet works, like it's easy to go one way and then you crank the other way and you keep cranking in the same direction. And in this case, the direction is getting worse and worse. So the fitness of the entire population is decreasing over time because of these mutations. Basically, there's no way to get rid of them if they become fixed in a population by chance. And Mueller's ratchet is definitely important in small populations, because the probability that a harmful mutation will be fixed just due to random genetic drift is very high in small populations. Okay, so let's consider our starfish carrying our allele for sort of normal body shape with stumpy arms and square tips on the same chromosome. Okay, so one good allele, two bad alleles on the same chromosome. Okay, even if it has a beneficial mutation, a rare beneficial mutation called big strong body, so it's twice the size of what it was before, guess what? You're still stuck with these two other mutations on the same chromosome if you're an asexual organism, because you can't swap them out through recombination. Okay, so now this beneficial allele is basically trapped on this chromosome with other bad alleles. Actually, this is a beneficial allele, but it's trapped on a chromosome with bad alleles, and it can't escape them. So even if you do have some fitness benefit from being twice the size of other individuals in the population, you still have these crummy alleles to deal with, and you have no way of getting rid of them. And that's what we call background trapping. Okay? So even this helpful mutation is trapped in a genetic background of deleterious or harmful mutations. Okay, so comparing the advantages and disadvantages of asexual reproduction, pros and cons. So the advantages for asexual reproduction: well-adapted individuals can clone their good allelic combinations. One major disadvantage is Mueller's ratchet. Bad mutations keep piling on every generation, and you have no way to get rid of them other than to die. Asexual reproduction has a twofold advantage in reproduction. You don't waste effort making males. But another disadvantage is background trapping. So even if you have a new beneficial mutation, it often gets trapped with harmful mutations on the same chromosome, or maladaptive alleles, as we call them. Okay, so let's finish up here. This will be a quick lecture. Do a quicker question. We can talk about this later. Okay.